So Andrea did a thesis in my lab looking at the effects of gestational opioid exposure on adolescent learning and memory, which was super exciting. She also worked on a collaboration with a collaborator of mine who's interested in chronic stress and inflammation. And now she's transitioned into industry and is going to tell us a little bit more about her experience there.
Thank you, Professor Robinson. And I'm very grateful to you for all your guidance. I wouldn't be where I am without Professor Robinson and a lot of the professors here in this room. Thank you all. So I want to chat with you today about discovery in biotechnology, which is what I currently do. I'm a senior research associate at Vertex Pharmaceuticals in Boston, and I am an MRA candidate, a Master in Regulatory Affairs candidate at the University of Pennsylvania. But before I start, I want to talk a little bit about myself. I'm from Mexico and moved to Massachusetts for college. Here you see a photo with my sister. I love spending time with my family. I am now a cat's parent. I love returning to Pine Cabo whenever possible. This was actually a few months ago. I love traveling with my mom and traveling with Williams Class of 21 classmates that a lot of the professors here know. And with my job, I currently still get to do a lot of that. I get to travel and explore and keep learning. So I'm very happy with my life now. But I want to chat with you about how I got here. So at Williams, I was a biology major and a neuroscience and public health concentrator. I was a member of the Zimmerberg lab from day one. So I was researching perinatal stress on mice, and in the Robinson lab, like Professor Robinson already mentioned, I was looking at adolescent opioid exposure and cognition and memory. So these were very exciting and mouse-heavy experiences. I came in knowing how to handle mice, but I think I was a little bit uncomfortable. I came in knowing basic study design, but still learned everything about planning, about researching from Professor Zimmerberg and Professor Robinson. So I wouldn't be where I am, which is literally designing studies and running studies every day without them. While at Williams, I did a few programs, and I want to chat about these in case anyone is interested in them. I did the UCLA Summer Health Professions Program, SHPEP, and it's a program that was at UCLA. I was able to shadow physicians there, dentists, and nurses. So you get to learn about their work with a public health angle, and you get to decide whether that's the career path that you want to take. I also did a Pima County Health Department internship. I was very interested in particulate matter. So this was a hands-on public health experience. I developed a statistical model for them to analyze the impact of the programs. I did a lot of tabling and visiting different community organizations. And I also did a Wadsworth Center REU, so the research experiences for undergrads. And here I was doing molecular research on diesel exhaust particulate matter. So always very interested in development and in the environment and how that impacts us as we grow. And the one thing that I also did that is typically very hard for biology majors with concentrations to study abroad. So I did the SIT Global Health and Development Policy program in Switzerland. And in this program, we learned a lot about different healthcare systems, and we looked at it from a lens of education, of healthcare, and we talked with industry leaders, with different agencies, the UN, with economists, and I planned a project. This was actually my first interaction with the pharmaceutical industry. I had no exposure to it during Williams until I think my third year. So I did a study that was looking at pricing in the UK, and I learned about the differences between pharmaceutical pricing in the United Kingdom and Switzerland. And I spoke to economists, and I spoke to different providers, and I got to see their perspective. So I think that was really an early sign of my interest today. And again, sorry, I forgot to mention, but if anyone has any questions, please stop me and I'm happy to chat about them. But after Williams, I joined the Yang Lab at Harvard School of Dental Medicine. I was a research assistant in developmental biology, and I investigated the role of cell signaling in the hippo-YAPTAZ signaling pathway. in embryonic development and mammalian anatomy and physiology using animal models of bone diseases. So a lot of mouse work, a lot of genetics. And this experience, I think, taught me a lot as well. I had done a lot of histology, a lot of study planning, a lot of literature searches, but this experience also made me do that at a greater scale. I don't know if that's the shared experience with the other speakers, but if you work as a research assistant, you do a lot, you wear a lot of the hats. So you are running a lot of histology, you're also helping sometimes with colony management, with study design. So you are really getting hands-on everywhere and touching a lot of projects. After that, well, at this point, I was considering dental school. And I considered dental school for a long time throughout my Williams career. And I thought a lot about what I wanted to do, dental school, continue researching, or perhaps explore careers that still touched science, but not in the conventional ways that I had been exposed to. So I applied to different positions in industry because Boston is a big industry city. And I kept hearing about all these new medications that were being released. And I was just always interested in seeing, like, what is the process? How do we get from something in the lab to something that touches people? And I wanted to be directly involved with that. So that led me to pursue a role as a senior research associate in pharmacology at Vertex, which I currently hold, and as an MRA candidate at UPenn. And I'm in my first year of that program. So I'm happy to chat about that, and I will tell you more about my job, my work at Vertex soon. But before I start, I want to introduce the drug development process because that's something that I had no idea existed, or I knew very little about the details when I was a student. And there are many ways in which students with a science background can integrate into that process. So this is my little lesson. So these are the phases of drug development. You have discovery, which is when you're finding a target and designing a drug. This is typically in the lab. It's a lot of in vitro and then sometimes a little bit of in vivo studies. And then you move to preclinical, which is where I currently work. I work in the middle of discovery and preclinical. So a little bit of this. And in preclinical, you're mainly looking at toxicity. So you want to make sure that the drug that you are providing to patients is not toxic and that it works. And we're also looking at pharmacokinetics, so what the body does to the drug, and pharmacodynamics, what the drug does to the body. And these are concepts that I learned in my physiology classes that I learned on paper and now get to see every day. So that's very exciting to me. I see what I learned in physiology, what I learned through my thesis, now take part in my day-to-day job, which is always very nice. I still pull my neural systems and circuits binder every now and then. Yeah, so what we're doing now is lead compound selection, which is companies will typically look at a lot of different compounds or so if you're looking at, let's say, medications, they'll look at different compounds and see the impact that they have on a disease. So it's hundreds of them sometimes, so that's where we're at. But then through my coursework, I don't have a lot of direct experience in the clinical phases, but I've learned a lot about phase 1 trials, which look at safety and tolerability, and they typically are done in healthy volunteers. And then I learned about phase 2 studies, which are dose finding and also continued safety. So now during phase 2, you move into patients, and then during phase 3, you move into continued efficacy, but also you weigh risk benefit. So you make sure that what you are offering patients is not only safe, efficacious, but is worth the hassle of receiving the medicine. And then you look to submit an NDA for small molecule drugs or a BLA for biologics to the FDA. And I wanted to chat with you all about the different ways in which someone with a science background or a neuroscience background could integrate into this drug development pathway. So you could either do things like I do in research directly in the discovery and preclinical stages, or you could advise clinical trials or be a project manager, or you could look at marketing. A lot of the marketing team, Vertex, my company, is very science focused. So everyone from discovery to marketing has a science background to an extent. So it's very exciting to be able to bridge two different worlds in which in any other context you wouldn't necessarily think about the science behind the medicine when bringing a product to market. So it's very exciting that you can really, the world is your oyster in terms of biotech. One thing that I wanted to also mention is that it's a non-linear process. So just because you work in discovery doesn't mean that the impacts of research in phase 1 or phase 2 trials will not come back to you and that you will have to continue contributing. So for me personally, working in discovery and preclinical has exposed me to a lot of the drug development pathway, because when things come back to us, if we see toxicity in patients, then you have to think about what is changing, what you can modify to make sure that the product is safe and efficacious for patients. So it's very it's a flexible process that responds to new knowledge. And I think that's different, and it responds to knowledge that may not come from you. So that's the biggest difference that I noticed from academia, is that in academia, for my thesis or for my research studies as a research associate, a research assistant, the data that I was generating was shaping how my project would go. In industry, it may be the data that someone else is generating in a completely different model that shapes your next steps. And I wanted to focus a little bit more on early drug discovery, which is what I do. So we look at drug screenings, we look at many targets, and then we narrow down what we think would work to target our disease. So we do this through in vitro, in vivo, ex vivo studies. We use animal and disease models, and we primarily evaluate safety and toxicology and work on IND applications. So my disease at work is Duchenne muscular dystrophy. So it's a chromosomal X-linked disorder, recessive disorder caused by mutations in the dystrophin gene. And some of these mutations lead to a premature stop codon. And it's characterized by the progressive and irreversible muscle weakness and deterioration. It has a prevalence of 1 to 4,500 male births, and it's the most common muscular dystrophy. It currently has no cure, and patients that have DMD experience a loss of ambulation and eventual decline in cardiac, respiratory, and muscle function. Therefore, there's a significant need for transformational therapies that target the genetic cause of DMD, because currently the approach is to receive steroids and improve the standard of living, or to foresee any challenges that patients with DMD would have, but they have very short life expectancies. And I wanted to chat a little bit about my research. This is an overview. But we aim to treat the underlying cause of disease. That's a big focus of Vertex. We know the cause of these DMD in these patients, the premature stop codon. And so we are using CRISPR gene editing technology to target certain mutations in the dystrophin gene that cause the disease. And we aim to deliver this cargo to muscle cells to potentially restore to near full-length dystrophin. And the concepts behind my research were things that I learned in my neural systems and circuits class. So again, I see what I learned at Williams every day with my work. Currently, we're looking to find an optimal, effective, and safe dose. And we conduct biodistribution studies, which are studies that look at where your drug goes. Are there any on-target, off-target effects? And we look at dose ranging. What is your target dose, and how will that translate to the human dose? Safety and toxicology. Are you seeing any effects in, say, things like germline editing? That's something that the FDA is very serious about, especially with genetic therapy. So it has ethical implications that are really seen in the day-to-day research. Currently, I design, lead, coordinate, and execute early discovery, hit to lead, IND-enabling preclinical studies in Vertex cell and genetic therapies. And again, my research is very collaboration heavy. So I work with the research teams, with the regulatory teams, with preclinical safety, and we work together to ensure that the studies that we are conducting day-to-day and designing align with what the FDA is requiring for approved drugs. So it's a very collaborative process that's guided largely by regulatory conversations and safety conversations. And this exposure through direct research exposed me through the field of regulatory affairs, which is what I'm currently studying. So through my current role, I have been able to draft pre-IND research reports, which are basically detailed summaries of experiments where we test our target product And we provide an analysis of things like stability, of target effects, we've done biodistributions, characterizations, and it has been very, it made me really question where these designs come from. So I wondered, so aside from the science that I'm doing every day, who is telling us what to do and why? And that's what really led me to regulatory affairs, is that I like understanding the bigger picture of the industry and how science connects to the world. And that's what I'm doing now with my master's degree. So I currently do consult FDA requirements and regulations for developing novel genetic therapies and conduct a lot of literature reviews, which I did while at Williams. And one thing that I do a lot of is competitive surveillance, and we all do at Vertex. So we make sure we read up on other products that are currently in the market, and we learn about their technology. So it's a field where everything is always changing, and it's not only the science that's changing, but it's also our regulation, and these regulations change from country to country, which I think has been very exciting for me to be able to bridge the science with the real world application. And in terms of lessons that I have from my current job, I sign up for everything, anything that teaches me about the pharmaceutical industry, about competitive landscapes for other pipelines. I signed up for, I'm not currently part of the paint program, although that would be really exciting. I would love to, but I go to all the paint talks. So I continue just learning about different departments, commercialization, marketing, and manufacturing, because it all ties together. A lot of even manufacturing, which you wouldn't think impacts research, it does. So even if you can do something in a petri dish, if you can't scale up or you can't deliver it in the same way every time, then it can't be a product that you can put in the market. So it's been interesting to learn about all these different stakeholders that work together to bring products to patients. Something else that I do is I try to take leadership roles. I am in the Vertex Ambassador program, so I promote inclusivity within Vertex, and it's something that I did a lot with Professor Chris Go and his program here at Williams, so I'm excited to continue doing that. I engaged with new recruits. I spoke to some Williams students that we had, some interns this year. I'm doing a short-term rotation in regulatory affairs, so I'm in a place where I need to be very proactive to learn about things that are beyond my immediate day-to-day. So I love learning about my research and the challenges and reading about what other companies are doing, but I also love learning about things that go outside of that scope. I also have a mentor, so that's something that I continue to do. I did that at Williams and I have at work. But yeah, I think my lesson from my current job is to be proactive and goal-driven, and that learning about the industry and about my projects is a goal in itself too. And I wanted to talk a little bit about my part-time MRA program. I currently work and receive funding from my company for this program, which is really nice. And they support me in attending classes while I'm at work, which is good. There's a good work-life balance. I learn about concepts like the drug development process that I just reviewed, but also I get to see it in day-to-day, so it's a very complementary system. And currently I'm learning to maximize compliance and minimize risk in the development of FDA-regulated products. And it helps me learn the foundation and application of science-based clinical investigation and corresponding regulations. And I learn from academic medical centers, private industry, and the FDA, which is exciting. And my next steps are continuing to grow within regulatory affairs, which is the interface between legal science and commercialization. So I'm trying not to broaden my curiosity too much, because once you see how much everything connects, I feel like it's good to always zone in on what your goals are. So yeah, I think my next steps may be more education and MBA, gigs rotations, and continued learning. Thank you. And I'm happy to take any questions.
Might be a silly question, but as you walk through sort of the day, a day in your life, what do you do each in your life? Do you find that it's, you say execute, do you hold pipettes? Are you in the lab? Is that maybe like a once a week working, or is it every day? Is it maybe once a month? Do you find that I know you sort of went into this to do research in a non-traditional sort of sense, but do you get to do like traditional research in a lab?
I personally do not most of the time. I think when I first started, it was a little bit more research heavy, and it really depends on the department that you work in. But a lot of my day is actually sitting down on my laptop and researching papers and developing research plans. But we work with a lot of contract research organizations, so you coordinate with them in terms of, we do conduct, so if something is high priority, very important, we all know how to do it, and we do it, but we also work with CRO. So it's an interesting transition from you doing the research yourself to having to trust other parties to conduct the research and still being responsible for making sure that every detail is accounted for. Yeah. So it's a little bit less. I think it's maybe like 75 sitting down, thinking about the research, planning it, and 25 sometimes doing a little bit of the research. Yes.
Andrea, you mentioned that there's a paying direct program, and for those of you who've taken neuroscience in the last couple of years, you know something about this drug. You know what channel drug is. Anybody? Maybe you can explain more about it.
I don't work within the paying program, so I do not.
It's the one that targets NAD 1.8, the mouse versus scorpion paper, for those of you who took neuroscience. Yeah. So you're all connected for
I was wondering if you could talk a little bit more about your strategy in getting this position. So once you realize that maybe you'd be interested in more pharmaceutical industry positions, how did you transition from your research assistant job into this?
Yeah, I think for me, I found a position that really aligned with the skills that I had, especially entry level. I think it can be a bit challenging to get into these positions, because I think one of the requirements for an entry level is that you have exposure to biotechnology, which is not possible. So I would find a position that really aligns with your skills, even if it's something that you've done once or twice in biology. Explain during your interview that it's something that you've done once or twice, but that you're happy to continue exploring. So just making sure that you list everything that you have done and putting your best foot out there, being confident, but also honest, and explaining that it may not be something that you do day to day, but that you're still comfortable with. And also internships. We've had a lot of pre-med students intern at Vertex, which is something that, at least in my graduating class, I didn't see. So that's also another opportunity to learn about it, perhaps from they also have doctors that work at the company as consultants and running the clinical trials and at different stages. So that's also a great opportunity to show them, like, hey, I interned maybe not within the medical department, but I interned with analytical development. And so I know how a pharmaceutical company runs. Can I get a job here? So I think something like that worked. Yes.
Sorry, is it fair to say that you do a lot of writing? Your job, it looks like this list involves a lot of writing, literature reviews, things like that. So I was curious, it's like how that compares to the writing in college classes.
Yeah, we definitely do a lot of writing and a lot of PowerPoint. So I would say the writing is more concise. It's not, typically you do have to read a lot and have very in-depth literature reviews, note papers, but when you do a write-up, say, of a study, or when you write the background for, or the rationale for certain parameters, then you have to be very direct. So I would say it's a lot of writing mini sources, but very concise. I wasn't used to not having like a 5-page summary of what I was doing. It's very, very direct, not exploratory. It's mainly you state the facts and you don't hypothesize. It's mainly what's happening live, why you're doing things, and not necessarily just thinking about things, not putting things in the air, because also anything you write can be used in court sometimes. So yeah, it has to be just the facts that what your data is telling you. Yeah. But I do love writing. My mentor is actually a medical writer at Vertex, and she's teaching me a lot about, not about medical writing, but about different parts of the industry. So I think everyone writes a lot from what I've seen.
Thank you very much.
Thank you.